Good morning, children. Today I am going to start with cell organelle lysosomes, and I give some homework from this topic of our last session, and I asked you to go through mitochondria and chloroplast also. Hope you have completed, and let's start with the lysosome now. Actually, lysosomes are called as suicidal bags of cells. Why? Because when a cell die or when the life cycle of cells got over, the lysosome consists of many different types of enzymes, and lysosomes burst and release these enzymes and digest the cells which lost its life. Okay. So, hence the lysosome destroy its own cell, it is called as suicidal back of the cell. Okay. So, lysosome were discovered by Christian de Dieu. These are known as suicidal bags. They are spherical bodies enclosed by single unit membrane. Actually, it is not like mitochondria, endoplasmic reticulum and chloroplast. It is not covered with double membrane. It is covered with only single membrane. Those organelles are covered with double membrane. They are spherical bodies enclosed by a single unit membrane. They are found in eukaryotic cell and absent in prokaryotic cell, that we know very well. Lysosomes are small vacuoles formed when small pieces of Golgi bodies are pinched off from its tubules. Actually, children, Golgi bodies you know very well. Endoplasmic reticulum as a cell organelle. At the end of endoplasmic reticulum, it pinch off and form a spherical bodies. Like that, many spherical bodies are pinched off from the endoplasmic reticulum, and all the spherical bodies together form Golgi apparatus. So, it is understood Golgi apparatus formed from endoplasmic reticulum. Next, from the Golgi apparatus, the lysosomes are formed. So, from endoplasmic reticulum, Golgi bodies are formed, and from the Golgi bodies, lysosomes are formed. So, that is what here discussed. Lysosomes are small vacuoles formed when small pieces of Golgi body are pinched off from its tubules. Okay, right. As I said, they contain a variety of hydrolytic enzymes that can digest material within the cell. So, inside the vacuole of lysosome, it consists of hydrolytic enzyme. This hydrolytic enzyme can digest materials within the cell. The membrane around lysosome prevent these enzymes from digesting the cell itself. Okay. So, the membrane around lysosome prevent these enzymes from digesting the cell itself. Since actually whether the cell is in live condition or its lifespan got over, what happened, the lysosomes contain enzyme and it burst and release the enzyme and digest of its own cell. Suppose if the cell is in live condition, what happen? If the cell is in live condition, if the lysosome burst and release the hydrolytic enzyme, and the live cells also dead. So, that is what it is protected by a membrane, and the enzymes are enclosed within that membrane and will not be released. Okay. Functions of lysosome, they digest intracellular digestion. Intracellular means what? Within the cell. Okay. They digest carbohydrates, protein and lipid present in the cytoplasm. Autophagy. During adverse condition, they digest their own cell organelles like mitochondria and endoplasmic reticulum. So, during adverse condition, what is called adverse condition? Maybe any scarcity of food and water. This or some other adverse condition, the cell organelle lysosome digest its own mitochondria and endoplasmic reticulum. And autolysis, lysosome causes self destruction of the cell. Then aging, lysosome have autolytic enzymes that disrupt intracellular molecules. So, what are the enzymes present here? Acid hydrolysis, nuclease, proteases, glycosidases, lipases, phosphatases, sulfatases and phospholipidases. So, these are the various enzymes this lysosome enclosed inside it. Okay. Next, phagocytosis. So, large cells are content or enveloped and digested by macrophages, thus forming a phagosome in cytoplasm. These phagosomes fuse with lysosome for further digestion. Actually, large cells are content or enveloped and digested by the macrophages. What are macrophages? Actually, macrophage is a type of WBC. It is a monocyte. It is a type of WBC. Monocyte are converted into a macrophage. The macrophage are large and irregular shaped cells. This macrophage can envelop the foreign particles, that is any pathogen or any other substance which are not belongs to our body are considered as what? Foreign particles. When enter into our body, these macrophages consider those microorganisms or are what it is microorganisms or any foreign particles and envelop it. Okay. So, when the macrophages envelop the cells, what happen? This forming a phagosomes in the cytoplasm. These phagosome fuse with the lysosome. Okay. Inside the macrophages, this this enveloped material form the phagosome. This phagosome fused with the lysosome and digest the whole structure of the enveloped material. Then exocytosis. Exo means what? Outside. Lysosome release their enzyme outside the cell to digest other cells. Okay, these are the various functions. Actually, lysosomes are very small organelles, but it perform various functions. Next one is microbodies. Eukaryotic cells contain many enzymes bearing membrane enclosed vesicles called microbodies. They are single unit membrane bound cell organelles, example peroxisome and glyoxysome. So, DAS and DAS consider as microbodies. Peroxisome and glyoxysomes are considered as microbodies. Actually, in eukaryotic cell, so it is understood it is not present in the prokaryotic cell, it is present in eukaryotic cell only. Enzyme bearing membranes which encloses the vesicles. Okay. Actually, vesicles means it is a bag like structure. Inside the bag, the enzymes are present, and this structure is called microbodies. They are single unit membrane bound cell organelles. Example, that means what? It is also covered with only one membrane, that is single unit membrane bound organelle. It is called, not double membrane bound organelle. Okay. Peroxisome, you could see the structure. It has a lipid plasma membrane. The plasma membrane is made up of lipid bilayer, and inside the plasma membrane crystalline is present. Peroxisome, first is organelle identified Christian D. Dew. Peroxisome are small spherical bodies and a single membrane bound organelle. It takes part in photorespiration and associated with glycolate metabolism. In plants, leaf cells have many peroxisomes. It is also commonly found in liver and kidney of mammals. These are also found in cells of protozoa and yeast. Okay. Peroxisomes were identified as cell organelle by Christian D. Dew. Peroxisome are small spherical bodies, and it also surrounded by single membrane only. Its function is take part in photorespiration and associated with glycolate metabolism. In plant, leaf cells have many peroxisome. When we see the plant, it consists of many peroxisome. It is also commonly found in liver and kidney of mammals. These are also found in cells of protozoa and yeast also. Next, glyoxysome, discovered by Harry Beavers. It is a single membrane bound organelle. It is subcellular organelle, contain enzymes of glyoxylate pathway. Beta oxidation of fatty acid occurs in glyoxysomes of germinating seeds, example castor seeds. Okay. Next, sparosomes. It is also spherical in shape, enclosed by a single unit membrane. Example, it storage of fat in the endosperm cells of the oil seeds. Next, we are going to see a centriole. Centriole consists of 9 triplet peripheral fibril made up of tubulin. The central part of the centriole is called hub, is connected to the tubules of the peripheral triplet by a radial spoke 9+0 pattern. So that means in the center there is no any other fibrils are present. So it is called as 9+0 pattern. The centriole form the basal body of cilia or flagella and spindle fibers which form the spindle apparatus in animals. Okay, actually this centriole is present only in the animal cell. Let's see, yeah, it's there, the structure is there, you see. Here is the structure given, children. You see please, cross section of centriole you please see. In that you get three ball like structures unite to form one unit. Okay, like that you can find 9 units around the center floral like structure. சென்ட்ரலில் ஒரு ஃப்ளாரல் ஸ்ட்ரக்சர் இருக்குது, அதை சுற்றி பார்த்தீங்கன்னா உங்களுக்கு if you count you could get 9 triplet unit. Okay, these are called what? Microtubules. These are called what? Microtubules. So when we see the cross section of, when we see the cross section of centriole, we could see 9 triplets units of fibrils on the periphery. Periphery means what? Outer region. In the center we cannot see any fibers in the center. It is the floral like structure is called core region. So centriole consists of outer 9 peripheral structure and also center core region. The center core region connected with the peripheral by a radial spoke. Okay, and it is present in the animal and animal cells and form a centriole during the fibers, spindle fibers during the cell division. The membrane is absent in centriole. Centriole is non-membranous organelle. பார்த்துக்கோங்க, centriole does not possess any membrane around it. இதுக்கு முன்னாடி நம்ம பார்த்தது glyoxysome, peroxisome, lysosomes and ribosomes all are covered with a single membrane, but here this centriole does not have any membrane around it. Okay, then endoplasmic reticulum, mitochondria, chloroplast, they are surrounded with 2 membranes. Ribosome, lysosome, glyoxysome, all the somes they are surrounded with a single membrane, but we cannot see any membrane around the centriole. So it is a non-membrane organelles. Next to vacuoles, actually it is present only in the plant cell. In plant cell, vacuoles are large, bounded by a single unit membrane called tonoplast. The vacuoles contain cell sap, which is solution of sugars, amino acids, mineral salts, waste, chemical and anthocyanin pigment. Beetroot cells contain anthocyanin pigment in their vacuole. That's what pigment means, it give color. Since beetroot contain anthocyanin pigment, it looks like a color, and vacuoles accumulate a product like tannins. So inside the vacuoles some products are accumulated in some plants and like tannins. The osmotic expansion of a cell kept in water is chiefly regulated by vacuole, and the water enters the vacuole by osmosis. Okay, what is the function of, actually this vacuole is present only in the plant cell, it is not present in animal. If present, it is very small in structure. And but when we see the plant cell, the vacuoles are larger than nucleus. It occupy the major region of the plant cell, and the major function of plant vacuole to maintain water pressure, which is known as turgor pressure. So the main function is maintaining the turgor pressure, which maintains the plant structure. Vacuoles organizes itself into a storage sequestration compartment. Example, vacuole store most of the sucrose of the cell, sugar in sugar beet and sugar cane. Then malic acid, what are the structures stored, what are the things stored in various plants of vacuoles, we will see. Sugar in sugar beet and sugar cane, the vacuole store sugar. Like in apple, the vacuole store malic acid. In citrus fruit, it store acid. And flavonoid pigments, cyanidin, 3 rutinosides in the petals of antirrhinum. So these are the various sucroses or acids found in the vacuoles of various plants. Next we are going to see about nucleus. With the nucleus we could wind up the class today. After nucleus we have a chromosome. After chromosome Okay, paravala. Okay, nucleus. Nucleus is important unit of cell. Let's see the full structure here. Yes, children, along with the structure we could go. Okay, nucleus is an important unit of cell which control all activity of the cell. It is very important. Okay, cell is surrounded by plasma membrane. Inside the plasma membrane, protoplasm is present, and it is differentiated in nucleoplasm and cytoplasm. In the cytoplasm, we have many cell organelles, whether it may be double membrane or single membrane or non-membrane bound organelles, and many structures are present inside the cell, many functions also taken place inside the cells. So, all the activity of the cell is under the control of nucleus. So, that is what it is called as controlling center of the cell. Nucleus holds the hereditary information. It is the largest among all cell organelles. It may be spherical shape or cuboidal shape or ellipsoidal or discoidal in shape, that depend upon the cell may be. It is surrounded by double membrane structure called nuclear envelope. Actually, nucleus also surrounded by double membrane, and this is called nuclear envelope, which has inner and outer membrane. The two membrane of the nucleus are called outer membrane and inner membrane. The inner membrane is smooth without ribosome, and the outer membrane is rough by the presence of ribosomes, and it continues with irregular and infrequent intervals with the endoplasmic reticulum. The membrane is perforated by a pores known as nuclear pores, which allow materials such as mRNA, ribosomal unit, proteins and other macromolecules to pass in and out of the nucleus. Actually, it is shown in the here, nucleo envelope, nuclear lamella. Okay, it is not shown here. Actually, nucleus is surrounded by double wall membrane, yes, outer membrane, inner membrane. At a regular or infrequent interval, not a frequent interval, infrequent interval, there are some pores with a interval of endoplasmic reticulum. So, this membranous is perforated by pores. Some pores means what? Holes. These holes are known as what? Nuclear pores. Through these pores, that means through these holes, materials are entered inside the cell and leave out inside the nucleoplasm and outside the nucleoplasm to the cytoplasm. What are the materials can be passed through this mRNA, ribosomal unit, proteins and other macromolecules to pass in and out of the nucleus. The pores enclosed by circular structure called annuli. So, sometimes when the pores are not in use, it is enclosed by a circular structure called annuli. The pores and annuli forms the pore complex. The space between the two membrane is called perinuclear space. Next, nuclear spaces is filled with nucleoplasm. A gelatinous matrix has uncondensed chromatin network and a conspicuous nucleolase or seen. So, then the chromatin network is an uncoiled, indistinct and remain thread-like during the interphase. It has little amount of RNA and DNA bound to histone protein in eukaryotic cell. Okay. The chromatin network is an uncoiled, indistinct and remain thread-like, okay, during the interphase. Actually, the chromatin reticulum only forms the structure called chromosomes during the metaphase stage of cell division. But during the interphase, there are various stages we can see in the cell division. One of the stage is called interphase. In the interphase stage, the chromatins or form a network structure which is uncoiled, indistinct and thread-like structure. And this chromatin reticulum consists of little RNA and more DNA bound to histone protein in eukaryotic cells. Okay, right. Next, during cell division, chromatin is condensed into an organized form called chromosome. Okay, as I said during the cell division, you know very well what is mean by cell division. That is one cell divided into two cells. So that is called what? Cell division. During cell division, chromatin, that is network of uncoiled thread-like chromatin reticulum, are condensed to form an organized structure called chromosomes. The portion an eukaryotic chromosome which is transcribed into mRNA contains active genes that are nautically condensed during interphase is called chromatin. The portion of the eukaryotic chromosome that is not transcribed into mRNA which remain condensed during interphase and stain intensely is called heterochromatin. Actually, this euchromatin and heterochromatin we can speak in detail in our 12th standard. That is, in this eukaryotic chromosome there are two different units, one is euchromatin, another one is heterochromatin. Euchromatins are it's can transcribed into mRNA, heterochromatins are not transcribed into mRNA and thickly stained. What is mean by transcribed, what is mean by mRNA, all we will detailedly discussed in our 12th standard. Okay. Nucleolus is a small dense spherical structure either present singly or in multiple inside the nucleus, and it is also not bounded by membrane. Like centriole, nucleolus also not bounded by any membrane. Nucleolus possess genes for rRNA, please note it. Nucleoli also possess subgenes. Actually, genes are located inside the chromosomes only, but apart from that some genes are present in the mitochondria, some genes are present in the chloroplast, and some genes are present in the nucleoli also. The genes which are present in the nucleoli possess for rRNA and tRNA formation. Okay. Next, functions of nucleus: controlling all cellular activities, that you know very well, and storing the genetic or hereditary information, coding the information from DNA for the production of enzymes and proteins. DNA duplication and transcription takes place in the nucleus. In nucleolus, ribosome biogenesis also takes place. And children, about chromosome structure, already 10th standard you had completed. Since you completed 10th standard, please structure of chromosome you go through by yourself. Suppose there is any doubt, you could ask with me. Okay. And next, special type of chromosome. Actually, chromosomes are present inside the nucleus, that you know very well. Inside the nucleus also, during the interphase stage of cell division, it is in the form of thread like structure. Okay, it is in the form of thread, but during the metaphase stage it form a chromosome, an organized form. So there are some special type of chromosomes are observed by some scientists in some of the organisms. Okay, not in all the organisms, in some of the organisms those structures are called special types of chromosomes. So these chromosomes are larger in size and are called giant chromosomes in certain plants, and they are found in the suspensors of the embryo. The polytene chromosome and Lamprey chromosome occur in animals and are also called as giant chromosomes. Polytene chromosomes observed in the salivary, where the polytene chromosomes are observed in the salivary glands of Drosophila, that is fruit fly, by E.G. Balbiani in 1881. In larvae of many flies, midges, that is Diphtheria larva, and some insect, the interphase chromosomes duplicate and reduplicate without nuclear division. A single chromosome with its which is present in multiple copies form a structure called polytene chromosome, which can be seen in light microscope. They are genetically active. There is a distinct alternative dark bands and light bands we can see in the polytene chromosomes, and about 95% of DNA present in bands and 5% in interbands. The polytene chromosome has extremely larger puff called Balbiani rings, which is seen in Chironomus larva. It is also known as chromosomal puff. Puffing of bands or the sites of indents RNA synthesis. As this chromosome occurs in the salivary gland, it is known as salivary gland chromosome. Gene expression, transcription of genes and RNA synthesis occurs in the bands along with polytene chromosomes. Same way, another one type is Lamprey chromosome. Lamprey chromosome occurs at the diploidine stage of first meiotic prophase in oocytes of an animal salamander and in giant nucleus of the unicellular algae Acetabularia. It was first observed by Fleming in 1882. The highly condensed chromosome forms the chromosomal axis from which lateral loops of DNA extend as a result of indents RNA synthesis. Next, flagella. Flagella I will discuss with you in my next session, children. And today's homework is explain the structure and function of nucleus alone. Okay. And I asked you to go through the structure of chromosome by your own. These two are the homework, complete and submit. No need to submit this because it is too large. So, but you complete and keep. And next class I have discussed about flagella. Okay. Bye children. Take care.